right back to it. So my name is Duncan Brake. I am one of the media directors for Sharks for Kids. If you guys haven't checked out the website, which you probably have already, as well as the webinars, we've got lots of cool educational resources and things to do for teachers and kids, and especially at home at the moment, with everybody being stuck at home online educating. Also too, if you're interested in watching some of the other webinars, check out www.sharksforkids.com/webinars. We've got some great scientists, conservationists, photographers, all sorts of people with Sharky goodness on there coming up soon. And also some of our webinars have been recorded. So if you want to see what we have previously, go on there and you can click on there and it'll take you to our YouTube channel where you can follow some of the people that have actually spoken already. So, as I mentioned before, we've disabled the chat and we have the question and answers section available. So if you have some questions for Vital, ask them through there. If I feel like I can answer them, I'm gonna be jotting them down on the way. If not, I'm gonna be collecting them and we will quiz Vital at the very end of the talk. All right, so I've known Vital for quite a while now. He's probably dove with great hammerhead sharks more than anybody I actually know, basically because he was doing his PhD studying them and was out there diving underwater with them every single day. Has a massive amount of time underwater with these animals, knows them all very well and individually, and he's going to tell you a little bit about the exciting research and projects he's been working on and listening to and studying for his PhD as well. So usually based in Bimini, originally from Switzerland, and I'm going to let him introduce himself, tell you a little bit more, and put his talk on and give you a little bit about his research. Perfect.
All right, thank you very much. I tried to share the screen now.
Mm-hmm. That's it. Perfect.
That looks all right on your side?
Yep.
Awesome. Cool. All right. Yeah, thank you very much for the introduction, Duncan. I figured I will dive straight in and tell you a little bit more about how we study the great hammerhead sharks in the US Atlantic and the Caribbean. So a tiny little bit of background first. Let me see if I can move that a little bit up here. There you go, that should work. So basically when we're talking about hammerhead sharks, we're not talking about like one particular species, but we're talking about actually 9 species of sharks that are commonly referred to as hammerhead sharks as of today. They're all part of the family Sphyrnidae. There are 2 genus in this family. So we have the genus Sphyrna that has 8 species in it, and then we have the genus Eusphyra that has only 1 species in it, which is the winghead shark. So there's a lot of research done on the different species. For me, I've been very fortunate to spend the last couple of years studying the great hammerhead shark. So that's what I will kind of like focus my talk on today. So to give you a little bit of a background about the species itself, the great hammerhead shark is a large coastal pelagic and semi-oceanic shark. So that means it has been seen very close to shore, but it's also seen further offshore, over continental shelves, and even well into the pelagic zone sometimes. It shows a circumtropical distribution. So what that means, you can see if you look on the right side of the slide, you see a world map and you see like dark blue areas. So basically everywhere you can see blue on that map, that's where great hammerhead sharks have been seen or recorded or documented. So when they're born, they're actually not that big. So they're between 50 and 70 centimeter at birth. So like around 2 feet, but they can grow up to 6 meters. So that's a large shark. Unfortunately today, like these really large or really long lengths are no more seen. Like hammerhead sharks above 4 meters have become extremely rare. They have to grow to a certain size before they are sexually mature or can reproduce. In males, that's between like 2.3 to 2.7 meters. In females, they tend to be a little bit larger. They're between 2.5 and 3 meters. So hammerhead sharks are live bearing, and after like approximately 11 months of pregnancy, they can give birth to anywhere between 6 to 42 pups per litter. I'm going to say like they're live bearing, so that means they give birth to live young. When we look to the, or like when we kind of like focus on the Bahamas, encounters with great hammerhead sharks historically have been rather rare. And if they have been seen, it was mainly when people would observe them coming into the shallow waters to hunt. So as in that paper from 1990, from which I used that figure, this paper by Strong et al. Described actually a predation in Bimini or close to Bimini, where they documented how a hammerhead can use their wide-shaped head to pin down and kind of like pivot on a ray, and then to get a piece out of that ray, so to predate on the southern stingrays we have in Bimini. And then more recently, there have been pretty reliable encounters in Bimini itself. And that kind of led to the Bimini Biological Field Station, or the Shark Lab, starting a long-term project back in 2010. And if you're interested in reading the first paper of that project, it's the Guttridge et al. 2017 paper, so it's open access. If you type in the title you see here into Google, it should come up with a PDF. I would like to share some of the findings from that research with you today. So the first thing in that study that we found is that great hammerhead sharks are seasonal residents in Bimini. So that means they're not in Bimini or around Bimini all year round, but they're only in Bimini during the winter months. So you can see that pretty nicely with that plot. So you have in like those black bars represent a residency index. So that's basically a measure that tells you how frequently hammerhead sharks have been detected in the waters around Bimini. On the bottom axis, you have the month, and then you have kind of like that red dashed line on the screen that tells you the temperature. So first off, to walk you through, you see that from December to April, that residency index is really high. And that's when we have reliable encounters with these sharks. So that's what we call the hammerhead season. But then you can also see that when these black bars are high, the temperature is low. So basically, when the temperature is between 25 to 27 degrees, those residency indexes are high. And then once the temperature increases during the summer months, that's when the hammerhead sharks leave. So you could say that the temperature might be a cue for these hammerheads to initiate their seasonal migrations, and they do move a lot. So we also found that once these hammerhead sharks leave Bimini, they can cover quite a distance. So we had sharks that were kind of just, if you look on the left side of the slide in that plot, the black dashed arrows, we had sharks that were bouncing in between the Bahamas and the Florida Atlantic coast. Then as they came back, but we also had sharks, we had actually 2 females that went from Bimini all the way to Charleston. And then we even had 1 female, we call her Artemis, she's one of our smaller females we tagged. She went all the way off the coast of Virginia and then came back the next year. So that was over 3,000 kilometers a migration. So that's a pretty long distance. And I said she came back, and that's actually another thing that we found out. So if you check the graph on the right side of the slide, you see that we had animals that came back multiple times, or like over multiple years. So we had sharks, for example, like 21M, that is one of our smaller males. He was first identified back in 2009-2010 season, and he was with us all the way every year pretty much until the season 2016-17. But we even have sharks that, as I said, we identified first in 2010, like Atlas, the other small male we have, and we literally only just have been diving with him as of 1.5 months ago in Bimini. So as I said, I'm talking about different individuals and saying, or like telling you that we have seen the same animals come back. Now you might ask yourself, like, well, how do you actually know that it has been the same shark? And that's a very good question. So obviously one way you could do that is using trackers. There are different trackers like satellite trackers, acoustic trackers that you can use on sharks. But what also has helped us during that study is the ability to identify those sharks when we were swimming with them. So I said to ID sharks underwater, you can use different external body characteristics to do so. So to walk you through that plot, so one thing that I really like to look at is kind of like the trailing edge or the notch patterns in the trailing edge of the first dorsal fin. So you kind of can think of it like a fingerprint for humans, but for sharks. So that's normally a pretty reliable way to tell a shark apart. So I said, because those notches seem to stay rather constant over the years, and then you can say which animal it is. But also any other fin characteristic, like Persephone has a missing part of her second dorsal fin, Atlas has a somewhat damaged anal fin, and then Chaos has, rather than that pointy end that you know from like the upper lobe of a caudal fin, kind of has that rectangular attachment here. And then another thing like that you probably have heard of or learned about is that sharks have that counter shading. So they seem to be dark on the top and then have kind of like a light colored belly area, ventral side. For some sharks, these darker gray areas seem to kind of like reach all the way on the ventral side and can create what we call a ventral patterning. So like obviously there are sharks that just have a white belly, then you can't really use that to say who it is. But some sharks do have like gray spots in that white area, like Epic, that's one of our smaller females we have. She has a very distinct ventral patterning that helps us to know when she is around to ID her. But then also other sharks like Gaia, our second biggest female, has that pigmentation spot that we have shown to be consistent in its appearance since 2013 behind her left gill slits. And then obviously you can also use external tags like this National Marine Fishery tag. So if you get as close to a shark that you can even read the number on it, then obviously that makes it easy to ID a shark. And these sharks, I said, they have been coming back to Bimini. So that's what we call philopatry. So philopatry is a broad concept that describes a geospatial preference of an animal to a particular location. I said with these hammerheads, it's Bimini. And we have actually been keeping track of which sharks we see in Bimini, and I'm maintaining an ID catalog. Currently, there are 55 different great hammerhead sharks in there. I made a little brochure with like the most famous ones from Bimini, and you're free to download that one. It's on our website, biminisharklab.com. If you go in the current research tab, you should find it as a PDF, because like if you should ever have the chance to come to Bimini and even dive with these sharks, make sure that like you have that ID catalog with you, because then you can go in the water and try to ID those sharks yourself, which is a pretty cool thing to do underwater. And then we currently have two major projects that we work with great hammerhead sharks in Bimini. One I started back in December 2016, and that's still going on, where we look at the potential effects of dive tourism and provisioning on the behavior and local species of these animals. So the great hammerhead shark dive in Bimini is world famous. A lot of people are coming to Bimini to kind of like experience those sharks up close. Here you can see Robin, one of my good friends from Bimini, giving a little piece of fish to Nemesis, one of our most famous female hammerheads we have in Bimini. And then the other one is my PhD project that I started in summer 2018, where we look at the fishery interactions, the conservation management, but also the ecological role of hammerhead sharks in the US Atlantic and the Gulf of Mexico. So first, I would like to talk a little bit about the provisioning project. As you have learned from a webinar last week, shark diving tourism is actually very popular. And it can be seen or can even be used as a marine wildlife tourism attraction that helps the conservation of the target species, because it is an alternative, or it offers an alternative to exploitative resources, to the exploitative use of marine resources. So that means like it can boost the local economy. You can use it to raise awareness for the challenges or threats sharks are facing. Like I said, you can put in like educational aspects in these activities. And it also, like I said, makes people understand that if you use a shark that way, it's kind of like a reusable resource, because the same shark, so let's say Nemesis shows up every dive and every day people are paying to see Nemesis. But if you then, in contrast, would open your waters for fishing and like a shark would get fish, that's kind of like a single use. So that also makes governments understand that their sharks are worth more alive than if they would open their kind of like area for fishing activities, and then that helps to start kind of keep the, or get the talks going about conservation management of these species. But then also, if you're an operator, you're kind of facing a trade-off, because you have to guarantee your paying customers the encounter with the animals. So what a lot of people do, they use food attraction and or provisioning as a technique to attract these animals. And while feeding of some animals can be like the most normal thing of the world, once it starts including wildlife and is done for touristic purposes, it can become controversial, because feeding wildlife has been shown to have effects. I said, like, it was shown that some animals show a reduction in home range. Other animals like these white-tip reef sharks, they actually have been shown to change their daily activity patterns. So they're normally active at night, but then the dive tourism takes place during the day. So they actually started being more active during the day now, and we don't quite know, well, how could that translate into the health of these sharks if they now are active during the night, during the day, but don't really have any rest periods left. So I said it can get controversial. So we try to understand the role of provisioning, but also environmental factors, and try to document individual differences in the observed behavior of the great hammerhead sharks at the dive site in Bimini. The cool thing is, like, as I said, we're able to ID those sharks. So rather than having a data set for a group that is fed and a group that is unfed, we actually had a data set for Medusa, we had a data set for Nemesis, we had a data set for Amphitrite. So we really had like individual data sets that we could compare with each other. And then we tried to combine all those findings and compare them to the species of the animals to figure out how the species could be changed by tourism activities. For the study site, I said I'm mainly based in Bimini, which is 50 miles east of Miami, great island. If you ever have the chance to visit after this pandemic, do so. It's a lovely island. I really like being there. And we were very lucky for that project to collaborate with Neil Watson's Bimini Scuba Center, a local dive operator that has helped us enormously with this project, and without which the project would not have been possible. So like a huge thank you goes to Neil and his crew. So basically what I did, or how my days looked like, every morning I would get up and I would cut a lot of bait. It's a little bit messy, it's a little bit smelly, but it allowed us to then kind of like weigh all the pieces, and we knew exactly how much a piece weighs. So then I could just go on the dive and count how many pieces a shark ate. And then at the end of the day, I could sum up the piece numbers, multiply it with the average daily bait piece weight, and I knew exactly how much one particular shark ate during the day. And then to kind of put that into relation, I worked with two indices. So the first index I used was the provisioning index. So it was basically a way for me to have a value, how frequently does one particular animal visit the dive site or attend the dive. So I just divided the number of attendant dives of a shark by the total number of events. And then for the comparison to the movement data, because we didn't have that many sharks tagged and I wanted to have like a more representative sample size, I actually multiplied this provisioning index with the average bait uptake to get a feeding index. So I would have then shark that we called highly provisioned, moderately provisioned, or even control sharks. So to give you an idea how my field days looked like, so here you can see kind of like an underwater setup. You have the dive master or feeder in the middle. Behind the bait box, you have dive guests in the water, and you have myself sitting behind the line of divers. And for me then, as you can see, like those sharks come really close. Like the first one was Medusa, that was Amphitrite swimming by. So I could just note down, like here we see Epic, all right, she got a piece. I would note down she got a piece, but I also knew like when did the sharks arrive, when did the sharks leave. Not all of them spent all 2 hours or the whole 2 hours with us. Some of them were present for half an hour or even less. Some of them said like were present for the entire dive. I would note down if I see anything like special about their behavior that day. So out of a single dive, I could get quite a lot of information, which obviously was really great to do, and gave me a good understanding like how these sharks act at the dive site. And then to kind of like give you an idea how data could look like, so those are some data examples. So one thing I then wanted to do is like look at how these like how differently the sharks eat. How much bait do they consume? Do we have sharks that consume more, or do we have sharks that consume less? So here you see the individual would be on this bottom axis, and the bait uptake would be on the left side. So say we compare individual A with that would be individual E. So very easily then it would be possible for me to say like, all right, individual A on average eats a lot less than individual E, but also not only is the average in individual E higher, but also the highest values recorded in individual E are higher than the highest values recorded in individual A. And then I said for every single animal, I would have graphs that could look like this. So I have the dive on the bottom axis. So every day we were diving. And I have the bait uptake again on the left side. And then every day I would know how much that shark ate. So you see it's like a lot of bouncing up and down. Sometimes they eat a lot, sometimes they don't eat a lot. But for every single day, I then could calculate how much they ate, transform that in a caloric value, and then say like, well, would that shark have met, or did that shark meet its daily energy requirement just from eating at the dive site? And that I then can compare to how they move. So to track these sharks during that study, we use acoustic telemetry. So basically you have acoustic tags, they look like this. So these are those small sticks. They are pretty small so that we can directly implant them into the body cavity of a shark. So it's a little surgical procedure. We catch the shark and we put the shark into tonic immobility, and you see here, like I put a tag in a lemon shark to give you an idea how that looks like. And then these sharks are coded. So they ping at a frequency, on average, the ones we use for our projects, on average, they ping every 2 minutes. So then like that tag is in the shark. And if you're lucky and your shark swims within range of one of your underwater receivers that are needed to detect the tag, that shark, or like the shark swimming by, will be recorded by that receiver. So every shark then has a tag with a individual ID number, or like with like an ID number that only that animal has. So let's say that would be Nemesis swimming by an acoustic receiver. I would get a record on that receiver, that number 18950 swam by at that time of the day. And then every 6 months I go out and like you see those yellow spots. So those are all this kind of receivers placed around Bimini. Currently we have 65 out there. I would go out, download them, and then for the last 6 months I knew exactly where my sharks were at what time, which then obviously I can compare to the days they were eating, to days where they were eating a lot, to days they were eating not a lot, or like compare sharks that on average eat more than other sharks. And the cool thing is I even had sharks that we tagged, but they were never actually seen at the dive site. So that's what we call our control sharks. They're not true control sharks because obviously they don't show, like we can't just think that they would show completely unimpacted behavior, because obviously dive tourism is going on, but we felt that's a good control group to give us a little bit more of an idea. How would those sharks move around Bimini? How would they use the space naturally without the dive tourism going on? I said that's like still work in progress. So we're actually working on the first paper being hopefully released soon. So yeah, stay tuned. If it's out, then yeah, go have a look at it and read it once we're able to publish. And then I said in summer 2018, I started my doctorate in zoology and started a new project that basically is split in 2 parts. So one part is the fishery interaction part, where we try to find an adequate conservation management strategy for great and scalloped hammerhead sharks in the Gulf of Mexico and the US Atlantic by discussing the efficiency of potential time area closures. And then the second part is kind of like with the idea, what happens if we should not be able to better conserve hammerhead sharks or apex predatory sharks in general? What could like be consequences for other animals in ecosystems where these sharks swim through? So again, here a little bit of background information regarding the current threats hammerhead sharks are facing. So they are a target or bycatch species throughout their range. And as a result of that, substantial population declines are expected to have happened in many areas where the sharks live. They are unfortunately very popular in the international trade in shark fins. And then recently they have been listed as critically endangered by the IUCN Red List. So critically endangered, that's kind of like the last stop before being declared as extinct in the wild. So obviously that is very, very alarming. So there are management measures to protect sharks, and some of these management measures have been shown to be very promising for different shark species, like for example prohibited landings, catch quotas, and so on. But we have reason to believe that these measures as a standalone tool might not be effective enough for great hammerhead sharks, and that has mainly to do with their very high vulnerability to mortality during capture. So there is a study that looked at the US bottom longline fishery, and they found that up to 94% of hammerhead sharks have been dead before they were even landed. So that means even if a fisherman doesn't really target hammerhead sharks, targets something completely else, if he ends up with having hammerhead sharks as a bycatch, a lot of them are already dead before he could, or he or she could even release them. And then the other thing is, especially for the great hammerhead sharks, they have a high post-release mortality. So again, like even if, I said a hammerhead shark is caught either as bycatch or because it has been caught in recreational catch and release fishing activities, even if that shark is released alive, in 47% there is a, like the shark still can die post-release, so even like later, because just from the stress it has experienced during the capture process. So kind of like taking all that together led to this paper, the Dolby et al. Paper in 2017, to categorize hammerhead sharks as one of 7 priority groups that would benefit the most from a more adequate conservation management. And that's kind of like where MPAs come into play. So marine protected areas have become increasingly popular to reduce the fishery-related mortality. But MPAs do have limits, mainly in size, because sharks move, and they move a lot. Like you remember, we had hammerhead sharks that swim over 3,000 kilometers. So there's a chance that no matter how big you design an MPA, if you're dealing with a highly mobile or migratory species, that at some point that species will leave the protected waters and swim through waters where it could potentially be fished or caught again. So that's why if you want to design these areas, like you have to include detailed age and sex specific movement and habitat use data to make them efficient. But it's also a pretty big thing to close off a whole area. And as said, with those highly migratory species, another strategy that is discussed that could be efficient are time area closures. So that's kind of like you try to find out where are your sharks, at what time of the year, and to what extent do they overlap in these areas. As said, in like now if we're talking about sharks, for example, with fisheries, and then you try to figure out, would it be beneficial for the shark if we close up that area, that particular area, for that particular time of the year. And that's kind of what we try to do with our project. So basically, again, we have a study site in Bimini, where you can see the red pin, but I've been very lucky to also have some really kind and great collaborators that allow me to join them in, I said, on fishing trips in the Florida Keys, down here with the Green Key, sometimes even a little bit more out in the Gulf of Mexico or even South Carolina, because I said, like, these sharks move and they are in different areas during different time of the year. So that kind of like these collaborations help me to get access to these sharks during different times of the year. For the predator-prey study, I said we're based in Bimini. That is completely based in Bimini, where we have access to great hammerhead sharks, but also spotted eagle rays and southern stingrays. So first off, to do, or kind of like for the fishery interaction project, one thing that has become crucial for us is, as I said, to accurately track these sharks. And while we could do that with acoustic telemetry, like I explained to you, acoustic telemetry kind of has as a prerequisite that you have receivers that can detect your sharks, but obviously that might become difficult if your sharks swim in the middle of the ocean. So what we use for this project is satellite telemetry. So we basically use satellite tags or transmitters that we put on the fin of the shark. And this tag here, you see those little kind of like metal pieces here. So those are wet-dry sensors. So these sensors to recognize if the tag is wet, meaning if the shark is diving, or if the shark's fin comes out of the water and those sensors get dry, the tag realizes, all right, I'm out of the water, and it starts transmitting. And what it tries to do is connect to a satellite, which there are a lot nowadays. If the satellite gets the transmission, it will process it, it will store it, and I then can download the data from an online database, accessing it through my computer, download a data sheet, and you see down here, like I'll get, I know which shark has been at which location, at which time of the day. So to put those tags on sharks, obviously we have to catch them. So that's kind of like the first thing we need to do. Hammerheads are large, they're very, very powerful, and instead they're very sensitive to being caught. So take all this together, and the hammerhead capture can become pretty intense and hectic because we try to be as fast as possible. In Bimini, we have a bite to release limit of 15 minutes that we absolutely do not kind of like go over to not risk the health of the shark. But then once we have the shark secure next to the boat, you can see how that looks like. We put the tag right at the top of the fin, or you can see it here on a scalloped hammerhead. The tag here is black because it has anti-fouling paint on it. So we put anti-fouling paint on these tags, so there's no biofouling accumulating on the tags, like weighing down the fin or creating a huge drag on the fin. And then once that tag is on and we release the shark for the tag's battery life, we get data. Every time the shark surfaces, we get a data point. So here we tagged the shark in Bimini, swam to Grand Bahama, then all the way up to Virginia and started coming back down the US Atlantic coast. And as I said, then we can figure out like, well, where has our shark been and what time of the year? Where is it in the summer months? Where it is in the winter months? And we can overlay that with fishing data and then say like, all right, there is an overlap with the fishing. Here is an overlap with the fishing. Would then kind of like try to assess if these areas would be beneficial or like could be efficient as a time area closures to reduce the kind of like fishery related mortality of these sharks. For the predator-prey interaction study, we used the hammerheads and the southern stingrays and eagle rays in Bimini as a model. And you might ask, well, why do you use these species? The simple answer is because the great hammerhead sharks in Bimini, they just very, very much like the rays in Bimini. So you can see here, that's a pretty cool video that I've been sent by a friend a while back. He found it on social media. Unfortunately, I don't know who recorded the video. So if the person is watching that webinar today and recognizes their video, please make sure to reach out. We would love to give you credit for it, but also like talk to you about that encounter. And as said, like we have literature from Bimini that shows how hammerheads eat those rays. Like here you can see a great hammerhead shark eating a spotted eagle ray. So basically what we try to understand is what happens in a food web. So the food web in the marine kind of like, or the marine ecosystems is pretty complex. So you see like you have a lot of interactions between these different animals of the food web. You have like high level predators or apex predators, you have middle level predators and low level predators. So we know that if you remove those apex predators, that could potentially have influences on the lower levels. But as I said, the question is like, how do all these interactions change if you would remove that top level predator? And we decided to use the stingrays to try and figure out a little bit more about those changes, because they're called mesopredators. And these are supposed to be they play a very important link in connecting the low and the higher levels. So if you look at Bimini and really, really simplify that, I said that's not representative, but just to simplify it, we have the hammerheads on top, we have the rays in the middle, and then like kind of like the low level species, like we have primary producers, we have primary consumers, and so on. And we want to understand what happens in the summer months when the hammerhead sharks are absent compared to the winter months when we have the hammerheads, kind of like to simulate what would happen if hammerhead sharks would kind of like be removed from that system. So we try to understand how do all these interactions change if the hammerhead is missing. And you see, like, that's a very simplified version, but you already see how easy and how fast these interactions become very, very complex. So basically we sample rays. To sample rays, we need to catch them first. So we put them in a tub because they're a little bit easier to handle as sharks. And then once we have them in a tub, we can sample quite a good amount of data from them. Like here we take a thin clip, but we even have a waterproof ultrasound which we can use to ultrasound the rays. And I wanted to kind of like summarize the whole data collection a little bit, because there is a lot. So basically, apart from measuring the rays to get an idea if they're sexually mature, how big they are, and so on, we take quite a good amount of samples that we try to interpret and then compare between hammerhead season and non-hammerhead season to see how things could change. So the first thing we take is a muscle biopsy. So we sample muscle tissue, and that gives us an idea about the trophic position or the interactions that ray has in the food web. So like who is the ray interacting with, at what level in the food web is it? To some extent, you can get a little bit of a snapshot at the diet, like who is the ray consuming, what is its basal source of energy. And we can do that by comparing atoms. They come in different heavy forms, and the ratio of heavy to light atoms kind of gives us an idea about that. And the same thing you can do with blood. But now, like, blood has a different turnover rate of those atoms than muscle. So muscle is more of kind of like a midterm snapshot at those interactions, whereas blood is rather short term. So we can actually look at both, like who has the ray interacted with short term and who with midterm. The blood we also use to look at body conditions. So kind of like what happens with the rays during hammerhead season and during non-hammerhead season. Like, are they constantly stressed during hammerhead season because they constantly have to swim around to avoid getting eaten, having kind of like what I try to represent with a low battery here. Or in summer when they don't have to do that, is their kind of like body condition better? We can do that by looking at as molecules like this triglyceride in the bloodstream. We can also use the ultrasound to do that. We can look at the liver density kind of to see like how much energy or lipids there are stored and try to assess it through that. We use, again, active tracking, not active tracking, acoustic tracking, I'm sorry, to figure out where are those rays when the hammerheads are present. Perhaps they go kind of like inshore, into shallow waters to try and hide or to try to find refuge from the hammerheads. And then when the hammerheads leave, they move out and go to different areas. We also use activity loggers or accelerometers. So very similar to like all those kind of like fitness trackers people wear on their wrists at the moment, that give us an idea how much do rays move. So do they constantly swim during hammerhead season because they constantly have to avoid a predator? And then in summer when the hammerheads leave, do they start kind of like relaxing a little bit more, spending more time on the seabed, like kind of sitting there and don't have to swim that much? So we try to figure out like what could be, I said, effects of hammerheads being present or absent on their activity. And that is actually already it. I try to give you a little bit of a kind of like insight in what we do currently with the hammerheads in Bimini. Obviously, if you run a project, there's a lot of people that help you with and kind of like without them, the project just wouldn't be possible. Like I have a lot of great supervisors back home at my university, but also in Bimini, I have colleagues that help me and give me advice. Like, for example, I said, if they see that I do something wrong, they will tell me like, Vital, that might not be the best way. Why don't you do it that way? I have a great group of collaborators that allow me to go in their boats. I said that helped me with fishing data, that helped me with biological samples. We have the Scuba Center, Neil Watson's Bimini Scuba Center, who helps us with the provisioning study. We have, as I said, like funders that give us money to buy supplies and equipment, or even sponsor us with equipment. And in Bimini, we have a good group of staff and volunteers that help me every day. And that's also the last thing before I give back to Duncan. We have volunteers. They're very vital for my project because I can let them go and catch, for example, rays for me. And also, like, if you guys should ever be interested to kind of like do the first steps in marine science or shark biology, like we would love to have you as volunteers at the lab. So feel free to check out our website, go to the volunteer section and get in touch. And with that, I will hand back over to Duncan.
Perfect. Excellent. Thank you, Vital. I'll get you to stop sharing your screen in a moment, and we should be right back onto the chat section. Great. We've got a ton of questions to go through, and thanks for giving the talk. I learned a few things I actually didn't know about the hammers. I've been lucky enough to dive with them a bunch. They're kind of right on the doorstep in Bimini, so they're amazing, amazing creatures. Actually, one of my favorite sharks on the planet, and they were one of my favorite sharks before I started diving with them. And as Vital mentioned, if you guys want to get involved in the research, the Bimini Biological Field Station, also known very well worldwide known as Shark Lab, it was actually founded by Dr. Samuel Gruber, who unfortunately is not with us anymore. However, he was one of the pioneers of shark science and discovered a lot of really cool things about sharks and made this facility that's been one of the oldest research stations in the world, long-standing data sets for things like lemon sharks, and is a great place for you to get experience in the field. And if you want to be a volunteer, like Vital is saying, and you get to do everything, absolutely everything, right down to helping tag the sharks, all the way to building, cleaning the gear, helping keep the lab clean, but then you get to be in the water with these amazing creatures and interact with them and get hands-on science. They do volunteer opportunities for anything starting at a month, and then all the way through to a year or more. Some people actually never leave. I didn't really ever leave Bimini. And then they also have some amazing opportunities as well for research experiences, which is, it's kind of like a very short, condensed version. If you don't have the time that you can spend that whole time away, then you can just go for a week and get to do all the really kind of fun stuff. Yeah, have a look at their website and see a little bit more about what's going on there. So I'm going to hit you up with some of these questions, Vital, so hopefully you're ready. I've answered some of them as we were going through. First off, Sydney would like to know, why did you choose this job and why do you like sharks?
So yeah, I think it just started with a general fascination for sharks, which is probably kind of funny because I come from Switzerland and we're landlocked, but I just ended up watching all those documentaries and I was like, well, that's an amazing group of animals, like that just fascinated me. And at some point, like we were asked like kindergarten, like, well, what do you want to do? And I was like, well, I just want to go and study these sharks. I kind of just followed through with it. And yeah, I mean, for me personally, it's the best job in the world. Like I couldn't be any happier. So.
Perfect. Yeah. Yeah. I definitely, I don't know what attracts me to these big toothy critters, but I think I've had a bit of an infatuation with them since a very early age. So, okay. Pascal Lafleur has a question of, is Florida like a hammerhead paradise? It seems like Florida's like a hammerhead paradise. Why do you believe this is? Why are all these sharks kind of aggregated around Florida, maybe in the Bahamas as well, in your opinion?
Yeah, I mean, that's a really good question, like, because obviously we see those sharks going up and down the coast and they seem to come back down to Florida every kind of like once the water gets colder. So that's one thing that kind of, I said, those sharks do most likely follow that temperature in the water a little bit, but then there's also like a lot of research going in, like what could, as I said, like the temperature might just be a cue want to move from one spot to the next spot because like they might follow other species that they predate on. Like for example, in Florida we have like the annual blacktip migration that hammerhead sharks have seen to follow, and they have seen to predate on those blacktip sharks. So I said there is a whole lot of different factors coming in, from environmental factors, biotic factors, abiotic factors, prey abundance in one particular spot in one, I said at one point of the year. Unfortunately, at the moment, we don't know where the hammerhead sharks go and give birth, or where the great hammerhead sharks go and mate. But I said, obviously, all those different factors can play in to kind of like create those temporal hotspots of those hammerhead sharks over time. So yeah, I said like that's that's definitely something like of active research, trying to figure out like why are they, I said in Florida at that point of the year, why are they coming to Bimini during the winter months? So yeah, there are a lot of different variables and factors that play in there, but definitely a really good question. Yeah.
Cool. And we have a question from Hannah, age 8, who's been tuning in, just asking how rare is the hammerhead shark? I suppose, could you see that they are potentially endangered?
Sorry, there was like a little delay.
No worries. Hannah, age 8, was wondering how rare a hammerhead shark is. Is it pretty rare to see them? Obviously in Bimini, not so much.
Yeah, like so like that's a difficult question actually. Like I had somebody ask me like, what is the rarest hammerhead shark in the past? I'm like, well, that is very different. Like obviously it depends like what factors you're looking at, Hannah. So like could be like if you're comparing numbers, but then it could also be like just comparing like where could you actually see them. Like I said, there are sharks, there are a species of hammerhead sharks that are literally only found in one place in the Carolinas in the US. With great hammerhead sharks, what we know is like they're solitary. So in general, these sharks travel alone. So that already kind of like makes it hard to encounter them. I said like we kind of know a little bit about their migrations at the moment, so we can go and see them in different areas. In Bimini, as Duncan said, we're very spoiled. We know if when we go to the right spot during the right time of the year, we will see up to 9 of them, which is unbelievable. This year, I think the highest we had in a single day was 12 different individuals, so that's amazing. I said, but normally they're solitary, so it's very rare that you see them. You're pretty lucky if you come across one in the open ocean. As it goes for their kind of like status, they're critically endangered. So that means they are not doing too well. I said critically endangered means they're only one step away from being extinct in the wild, according to the IUCN Red List. So I said they have become pretty rare. Or are, as I said, like they were already, due to their solitary behavior, low in abundance and being apex predatory shark. But yeah, great hammerhead sharks are rare to encounter, in my opinion.
Cool. Yes, I know before we sort of kind of started seeing them in Bimini, they were quite very rare to see, like especially diving elsewhere around the world. I've been in a lot of oceans with my job, and I've very rarely seen great hammerhead sharks. Which actually, when you were saying that the great hammerhead is they're quite nomadic, lone individuals, that brings us on to a question from Caitlin, who asked if the great hammerhead sharks hunt in packs or alone. Is there any evidence pointing to one or the other?
To my knowledge, I'm not aware of any evidence that would point towards them hunting in packs. I said they're solitary. So like to my knowledge, like I said, they are pretty nomadic. Like I said, if it's like those groups we have in Bimini are artificially created through the baiting. But to my knowledge, I said there's no evidence yet that they would hunt in packs. And if I would have to answer your question, I would say like, yeah, they do hunt in a solitary mode. So yeah, that's
perfect. Cool. Awesome. Actually, got a gentleman or a guy called Kiefer. Kiefer has written in and is intrigued to know, do you have to have some sort of license or certification to tag a shark?
So like that depends which area in the world you are, which country you're operating out of. There are permits that you need to have. I said depending what species you're dealing with, they might be under different conservation acts or kind of like under different regulations, depending on how, I said, like what their conservation status is. So in Bimini, we do need a permit from the Bahamian government that allows us to go out and catch those sharks and tag them. So you can't just go out and catch a shark and take muscle from it, or go out and put a satellite tag on it. So we do need to have permits to do that kind of work.
Yeah, it's definitely it's something, especially with great hammerheads. I would not recommend you trying to be involved in tagging it unless you're working with a very experienced team. They obviously stress very easily. So you need to have a lot of experience tagging sharks, especially other species of sharks like nurse sharks and tigers, before you even want to even consider putting a tag in a great hammerhead. Cool. We've got Olivia has asked, is the big famous female hammerhead still alive? I think this might be referring to one of the ones that's been on all the TV shows. It turned up on, like, I think Monster Hammerhead, maybe on Shark Week and stuff, and it was actually, from being in Bimini in the Bahamas so many years, there was actually a great hammerhead they referred to as the Harbor Master, which was a giant hammerhead that used to come up into the harbor in Alistown in Bimini, and was seen for many, many years around hammerhead season. Now, I know that individual, we're not sure that individual was ever tagged or was around when the tagging project started. So it's hard to know if that exact individual is still around. However, Vital will probably tell you about the biggest female that he has tagged.
Yeah, so we have a couple pretty big females out there. The biggest one we call Amphitrite. She is 3.48 meters in total length as of the 13th of January 2017. So she might have grown a centimeter or two since then. But she's the biggest shark we currently have in our database. And she has been around for a long time. Like, if you remember that graph I had in the presentation with the yellow, red, and blue dots, like, she's one of those really early sharks that has been with us, and she has been with us for so like since 2013-14 season. I said it took us quite a while to tag her. She has been pretty elusive to our capture approaches, so we only tagged her back in 2017. The second biggest is Gaia, and she is kind of like the gentle giant, we call her, because like to dive with her is like one of the most relaxing things you can do to some extent, because she's super relaxed. She kind of glides in, she doesn't get stressed out at all, like different to Amphitrite. Amphitrite tends to rush in a little bit, but Gaia too, like, she has been with us since 2013. Actually, Gaia herself doesn't have an internal tag. So that's something we still try to do, but like, yeah, luckily those large hammerheads we had, they're still with us. We had sharks, I said, over time, like, because they do leave in summer, we never know which ones are coming back. So we had females or also males that did not come back the next year, which is obviously very, very sad for us. But yeah, so those big females, I said, I'm not sure if one of those was the one that you were referring to. I said they were called differently in different TV shows, but the two biggest females we ever had tagged in Bimini, they're still here.
Yeah, awesome. Yeah, and they are amazing sharks, those two big ladies Vital was talking about. I've had a lot of time in the water with them, and they definitely make a connection with you when you're underwater working with them. So we've got a poop question. It's always good. So be ready for this, Vital. We've got a poop question from, it's either Katie Smith or Judah, which might be using Katie Smith's account. So shark poop, obviously hammerhead poop, does it act as a fertilizer, do you believe? Do you believe, like, if a hammerhead poops, does it act as like a fertilizer or some sort of nutrition that helps other animals?
That's a very good question. I actually have never seen a hammerhead poop, but I did see nurse sharks poop in front of the bait box. And when that happens, normally, like, all the jacks, so you see on Duncan's background, there are like a couple pictures of hammerheads, and you see all those silvery fishes in it, so all those jacks. So yeah, exactly. So all those fishes, whenever a nurse shark decided that it had to go to the bathroom, those fishes would just run after it and eat the poop. So basically, it is very, like I said, like similar to terrestrial animals. Obviously, you take up nutrients, you break them down, and some of the nutrients come back out at the rear end. So if these come out, obviously those nutrients can still be used by other animals. I said, like lower level consumers, but also like producers potentially. So I said, like, I'm not an expert on shark poop, but it definitely, like, from what I observed in the wild, there is nutritional value still in it to some extent that can be used by other animals.
Perfect. Yeah, I've definitely seen other animals take advantage of shark poop before as well. And who doesn't love a poop question? Okay, we've got time for 3 last questions here. First off was Natasha was worried about sharks getting hurt on tourist dives. Scientists, I explained as well with like a lot of the tagging part. It's a little bit like an ear piercing with the cartilage, but do they ever get hurt on these tourist dives?
I said that's what makes shark diving or wildlife tourism in general controversial, because there is the potential for the animals getting hurt. That doesn't necessarily have to be through human actions. It can also be that you just create a very large aggregation of animals where the animals themselves become a little bit rallied up if they have to share the space or compete for food with other animals. So there is potential that these sharks like that there is a negative impact. Like there is a series of papers that discusses impacts at stingray provisioning sites. And they actually found that stingrays at the provisioning site had more bite marks from conspecifics. I said they kind of got more injuries from boats at the dive site. That kind of like they got hurt. Like that can even go as far as just parasites being kind of like shot up between the animals that can have an impact on the health of the animals. As in Bimini, I said that's something that I kind of can recommend. If you decide to do a wildlife tourism activity, make sure that you choose an operator that you know that treats an animal with respect. Like I said, there are operators out there where the dives can go a little bit into kind of like a Wild West dive. So that's something like if you decide to go out there, make sure that you find an operator that really cares about the animals, like where the animals comes first, that gives the animal the respect, and then obviously the risk of an animal being harmed can be reduced.
Yeah, that's a very good piece of advice for anywhere that you travel in the world. All right, slightly off on a bit of a tangent with this one. What is your favorite prehistoric shark, Vital? I know for me it's probably the Stephacensis, which has like this big old weird hammer on its head on the back of its neck up here. I've absolutely no idea how it would be used properly, but it's like with a big hammer grows out the back. But what about you? Do you have a favorite prehistoric shark?
I mean, there's definitely some really cool prehistoric animals out there. I think I wouldn't say it's my favorite per se, but I also don't know if I can even pronounce it properly. But the shark, the Helicopteron, like the one that had kind of like the saw.
Helicoprion, yeah, they're really cool.
There you go. I think
buzzsaw in their mouth. That's pretty bad.
Yeah, so like, and I say like, I wouldn't say it's my favorite, but I do like the discussions around it because like, obviously, like how would it be able to use that kind of saw without, it doesn't kind of look functional. And then also like an interesting, like, kind of thing is like, are we actually sure that that is not just an artifact of how that shark kind of got pushed into the sediments when we found it? Could it be that we might be interpreting that wrongly? So I think there's a lot of unanswered question about that shark that kind of interests me. Like I said, I wouldn't want to kind of put myself on a one favorite prehistoric shark, but I think that one is a very interesting to look at because there are like a couple questions that we still have to figure out about it.
Yeah, that's definitely a very cool one. And you guys should look that up. Google it, Helicoprion. It's a very cool looking shark. Apparently one of the very first as well. We also have, and we're going to conclude on quite a lovely question from Claire, aged 10, who was wondering, what can we do? What can anybody do to help sharks and help these great hammerhead sharks?
That's a great question. So like I said, and it's actually like, I really like the question because it kind of like shows like everybody can do something. Like those are the little steps, like tell your friends about what you know about hammerheads. Try to kind of, like I said, raise awareness about the threats these hammerhead sharks are facing. Make decisions that could potentially help these sharks. Like, for example, when it comes to seafood, that's like a big one. For example, like we have learned that hammerhead sharks end up end up not only as targeted like species in the commercial fisheries, but they often end up as bycatch and giving their high mortality. So like I said, if you make sensible choices about your seafood, make sure that you eat seafood that perhaps you even know how it was caught, or only, like I said, say like, okay, I eat fish, but only if I go out there myself and catch it myself. So try to make sustainable decisions about it. I said, like, it can be very little things that can potentially help these sharks, I said, for making decisions, but also kind of like try to encourage your friends, I said, in school, kindergarten, like whatever age, or like even if you tell it to adults, like even adults, like if you tell them like, hey, but why don't you make a better choice about your seafood, or why don't you like have you known that hammerheads are that sensitive to capture? So there's little things that you can do that definitely help to better protect these animals.
Yeah, no, it's an incredibly good point. And one of those things that is like, know what you're eating, and actually know what you're eating, because sometimes there'll be things on products like says contains white fish, which is an excuse for people to load things with shark. They did a lot of genetic testing on things from cat food all the way through to soup packets, which had like marine soups, like fish soups, which had all different bits of fish and stuff. And they actually found hammerhead shark, which was labeled as white fish in those soup packets as well. So definitely know what you are eating and be a responsible consumer. And even at the most basic level, try and minimize plastic out of your lives, because using a reusable water bottle instead of plastic water bottles all the time, those plastics and those microplastics end up in the ocean, and then they end up in the food chain, and they are ultimately going to be affecting these magnificent animals. Cool. So that pretty much concludes the webinar for today. However, keep an eye out. If you go to www.sharksforkids.com and you go to the education section, you'll be able to, if you've been watching a bunch of these, you'll be able to download your webinar certificates. Very exciting announcement as well. We're going to be putting a pretty cool hammerhead poster up there in the next day, where you'll be able to download that poster. It's going to have all the cool information. So that's on the education section of the website. And additionally, stay tuned, because at the end of the week, because we're all stuck at home here at the moment, and we can't get out into the open ocean environment. So at the end of the week, we're actually going to be putting out a virtual 360 shark dive with these great hammerhead sharks that you can jump on. It'll be available through the website, and you'll be able to, if you have a VR headset, you can watch it on your VR headset, or you can put it on your phone and move your phone around, and you'll be able to see what it's like being underwater with these amazing creatures and some of the other sharks that we have seen in the Caribbean. So keep an eye out for that. That'll be a very exciting announcement. Also, make sure you check out the Bimini Biological Field Station's website, Shark Lab, for more information about these cool projects. And if you want to help support Vital's current research, he may have some t-shirts available, so you can always hit him up there, which have some cool hammerhead shark designs and stuff, which will help him buy some tags and do some fun stuff like that. Cool. Alrighty. Well, thanks again for your time, Vital.
Thank you very much for having me.
You, bud. And I will be stopping record now, and I'll be closing.